DNA replication begins when the enzyme helicase unwinds two strands at the origin of the site or replication fork. Then, single-strand binding proteins bind to the two strands to prevent them from repairing. The new DNA strand is synthesized by the enzyme polymerase III. Because the DNA is anti-parallel, the elongation is different for the two strands. The leading strand is able to be continuous as it flows from the 5 to 3 direction. However, the lagging strand is elongated discontinuously since the 5 to 3 is opposite from the direction the helicase is unwinding the strands. Now, a short sequence of RNA nucleotides, also known as a primer, is synthesized by the enzyme primase. This allows for the new DNA strand to start from the 3' end of the RNA primer. The segments of the lagging strand are called Okazaki fragments. Now, the primers can be removed and replaced by DNA nucleotides, which is carried out by the enzyme polymerase I. In order to connect the Okazaki fragments, DNA ligase joins the sugar phosphate backbones to the Okazaki fragments into a continuous DNA strand.
Initiation begins with the enzyme RNA polymerase binding to the starting point of the gene, which is called the promoter. The promoter of the gene includes within it the transcription start point and commonly includes a Tata box. In eukaryotes, a collection of proteins called transcription factors mediate the binding of RNA polymerase, which is called the promoter. Only now can RNA polymerase II bind to the promoters. This whole complex of transcription factors in RNA polymerase II is called the transcription initiation complex. This causes the DNA double helix to unwind so that RNA polymerase can begin reading bases. RNA polymerase adds nucleotides to the DNA template strand in the 3 to 5' direction. For each nucleotide in the template, a complementary RNA nucleotide is added to the 3' end of the RNA strand. RNA polymerase will keep transcribing until it gets signaled to stop when it reaches a transcribed terminator. This causes the RNA polymerase to detach from the DNA. The new strand of mRNA is complete and detaches itself from the template strand of DNA.
mRNA contains exons that code for protein and introns, the non-coding section. Non-coding introns need to be removed, and a 5' cap and a 3' poly A tail need to be added. They are added to protect the transcript from being broken down by enzyme and to facilitate attachment of mRNA into the ribosome. The introns are removed by a process called RNA splicing. In this step, the small nuclear RNA in ribosome along with other proteins create a spliceosome to catalyze the excision of introns and joining of exons.
The tRNA carrying methionine attaches to the small ribosomal subunit. When the initial tRNA with the anticodon UAC binds with the start codon AUG, a large ribosomal subunit joins the small one to form a complete ribosome, and protein synthesis is initiated.
Once the matching tRNA has landed in the A site, it's time for the action. That is, the formation of the peptide bond that connects one amino acid to another. Termination is initiated by one of 3 stop codons into the ribosomal A site. Release factors trigger an enzyme that normally forms peptide bonds. They add a water molecule to the last amino acid of the chain. This reaction separates the chain from the tRNA, and the newly made protein is released and the small and large ribosomal units dissociate.
In the lac operon in bacteria, instead of having one gene, bacteria will have all the genes required to deal with lactose will be put right next to each other. Their names are lac, Z, Y, and A gene. But they each make a protein and they each help to break down lactose. Above that, they'll have a regulatory sequence called a promoter where RNA polymerase grabs on. And the other thing they'll have is an operon called an operator. A repressor will plug right into the operator, and as long as the repressor is available, RNA polymerase can't get on. When the repressor is here, the operator is now in the off position. In other words, we can't make these genes. But let's say lactose shows up. The lactose, you'll notice, is going to fit perfectly into that repressor, and when it fits in the repressor, it changes the conformation or the shape of the protein. In other words, now the repressor doesn't fit in the operator anymore. RNA polymerase now fits. RNA polymerase is now going to run down. It's going to make each of the mRNAs for each of those lac genes. each of those are going to make a protein, and each of those are going to break down the lactose. And so we can deal with the lactose and we can metabolize that lactose. So now the lactose is gone and the repressor is going back to that original shape. Negative control is seen in the trip operon. Instead of having just 3, it has 5 different genes arranged next to each other. It's actually off whenever the chemical is present. We need that tryptophan and amino acid to make proteins, and so bacteria, as long as tryptophan is present, they don't want to have to make tryptophan on their own. Without tryptophan, the repressor is going to change shape, which allows RNA polymerase on. RNA polymerase is going to quickly make those 5 proteins, and then those 5 proteins are going to make more tryptophan. So the tryptophan fits again, and it's going to turn off.